So let's get started. So we're going to start with tonicity, that concept. And tonicity deals with water movement, okay? Where water moves in or out of a cell. So I'm going to set up 3 beakers here, 3 glass beakers. 1, 2, 3. So this is my beaker number 1, beaker number 2, beaker number 3. Okay. And my 3 beakers are going to have 3 different solutions in them of water and salt. They're all going to contain water and salt, but different concentrations of each. Oh, someone's hold on. Okay. So I'm going to do, let's do. Okay. Beaker number 1 is going to have this water in it. Okay. 2 is going to also have water in it. 3 is going to have water in it. And beaker number 1 is going to have 99% water, 1% solute or salt. Or the solute is salt, NaCl. So I'm going to write 1% NaCl in beaker number 1. Dissolved in the water. And to sort of visually represent that, I'm just going to do a couple dots submerged in the water here. Beaker number 2, I'm going to put in a little more salt. I'm going to put in, let's do 5% salt. So this is a 5% salt solution. So there's going to be a couple more dots salt in my water here. And my last number third beaker here, I'll do 10% salt solution. So this is more dots, right? Higher concentration of salt. Notice I'm leaving a little area here blank. It's because I'm going to submerge a cell. It's not going to be drawn to scale. Of course, cells are microscopic, but to understand the concept here, I'm going to draw 3 cells in my beakers here. Actually, make it even bigger. So I submerge 3 cells into my 3 beakers here. Of varying salt solution concentration. And now I'm going to tell you that each one of my cells, well, we've learned that water is the universal solute of life. All life is submerged in water, dissolved in water. So cells, things that surround cells, it's all water with varying concentrations of solutes dissolved in that water. And so my cells are filled with water, but they also have salt. Okay, so I'm going to write that this cell, by the way, this is tonicity. If you want to, I forgot kind of the title here of this lecture. This is tonicity. All right. So I'm going to tell you that all of the cells have 5% salt solution, 5%. So this cell has 5% NaCl. This cell also has 5% NaCl, and this last one too. dissolved in the water of these cells. Okay. So the concept with tonicity is that when there is this sort of two separate states where you have an internal environment and an external environment, there's going to be a net movement of water. And it's going to depend on the relative differences between the internal environment and external environment. But there's a common theme. The common theme is that water moves toward the solute. So I'm going to write that down. It's one of my key phrases. Water moves towards the solute. So wherever the solute concentration is higher, water is going to move towards that solution. Okay. So there's a couple new words to learn here, but first let's just take a look at where water is going to move in each of the beakers. So in this beaker, our first beaker, the water is going to move toward the solute. It's going to move in the direction of where the solute is greater. And so water is going to move from outside of the cell in our first beaker. Why? Because the solute concentration is higher inside the cell. Water moves toward the solute. So I'm going to draw my arrows pointing into the cell. Water is going to enter our cell here. We call this situation hypotonic. The solution surrounding the cell is hypotonic to the cell. Hypo, kind of like hypothermia, less temperature when you're freezing, right? Hypotonic is referring to the solute. Outside of the cell is less, 1% versus 5. So it's hypotonic to the cell. Let's move on to this cell. Equal, right? Inside and out. We call this isotonic. And so in an isotonic state with the internal environment and external, water is going to move in and out of the cell at an equal rate. So there's no net gain or loss in either the solution surrounding or inside. So I'll draw an arrow going in and an arrow going out equally. This is isotonic. The solution is isotonic to the cell. Last, this is our hypertonic solution. It has a high hyper level of solute compared to the inside of the cell. Where's water going to move? In our third beaker. Exactly. It's going to move out of the cell. Totally. So I'm going to draw arrows going out. It's moving towards the solute, towards where there's more solute. So this cell, because it's losing water, actually starts to shrivel up. No good. It's dehydrating, it's losing water, right? This cell, no change. And this cell is going to start to enlarge, and it can actually burst, which can be problematic if a cell is placed in a hypotonic solution. So I'll give you a couple interesting sort of anecdotes here, but I watched a colleague teach tonicity once, and her sister is a nurse. And when she was in nursing school, so you practice giving injections a lot. You use it on dummies, you give each other injections. And the nurse's friend was practicing at home and used distilled water to inject herself and practice giving an IV. Distilled water has no solutes in it. It's filtered, it's pure water, 0%. Our cells have solutes in them. They have ions, they have minerals, they have salts. And she actually, her skin started to swell up and inflame, and she had to be rushed to the hospital because the distilled water she injected straight to her blood vessels, her blood cells, her skin cells, cells in her connective tissue, they were all blowing up and bursting, rupturing, yeah, and caused a huge bruising and all this stuff. Yeah, so it was kind of an important lesson, right? And why this is important. A great hypertonic example, this is why you never want to drink seawater, because although if you're stranded at sea, seawater is very, very salty, high salt concentration. And ironically, when you drink seawater, it actually dehydrates you further because it pulls all the water out of your cells that are less solute concentration than the high salt seawater, and you just pee it all out and you actually become more dehydrated. It's kind of crazy, but that's why. One more quick note I want to mention. You can talk about these different states, tonicity states, when you're talking about the solution surrounding the cell, but also the solution inside. So if I were to ask you in beaker 1, is the solution inside the cell hypertonic to the solution outside, isotonic, or hypotonic to the solution outside the cell? So far, we've only been talking about solution outside the cell. But what is the tonicity of inside the cell to outside? 5% to 1%. Yeah, it's hypertonic. So if I asked you and I said, fill in the blank, the solution inside the cell is blank to outside the cell. It's hypertonic inside the cell, has more solute. We saw, though, with this 1% surrounding, that the solution outside the cell is hypotonic to inside the cell. So it can be used in either, depending on the context of what you're asking. You can use these different words. Of course, isotonic, no matter which one you use, since they're the same, it would be isotonic to the cell or isotonic to the surrounding solution. So there's a couple hypotheses as to why water does this, but one of the prevailing hypotheses, and sometimes this helps students, we learned about how osmosis where water moves, goes from high concentration to low, right? That's actually what's going on. Outside the cell here is 99% water, right? If it's 1% salt. and the rest is water, it's 99. Inside the cell is 95% water. And so it is indeed going from high to low. Can move this up too. And then here, of course, the water concentration is the same inside and out. And then here, where the water leaves the cell, you have 90% water outside and 95 in. Therefore, you are still going from high to low. Osmosis is still taking place.